Hello everyone, this is Vincenzo Romano and I'm assistant professor at the Erasmus Medical Center Rotterdam. Today I'm gonna talk to you about a recent paper that we published on current biology, which is titled Olivocerebellar Control of Movement Symmetry. A wide range of motor behaviors such as walking, swimming, and flying requires the coordinated movements of the left and the right part of the body. However, how the brain achieve such a fine coordination of the left and right part of the body is not well known. And as most of you already know, one part of the brain that is essential to achieve fine motor coordination is the cerebellum. And one of the main source of inputs of the cerebellum is the inferior olive, which is a brain structure of the lower brain stem and provide the cerebellum and in particular its main neuronal type, which is called Purkinje-cel, with very powerful electrical signals. And this very strong excitation of Purkinje-cel is called complex-spikes and is relatively rarer to another type of electrical signal produced by the Purkinje cells, which are more frequent and are called symbol spikes. In this study, we recorded the symbol and the complex spike electrical activity of Purkinje cell of the cerebellar lobules called CRUS1 and CRUS2. And we also optogenetically manipulated the olivocerebellar input to artificially produce complex spikes. And we recorded the produced complex spikes while monitoring the mouse whisker movement with a camera, high-speed camera, and we also did anatomical tracing to investigate the anatomical connection between the left and the right cerebellar hemispheres. We found that complex spikes preferably occurred at the switching moment between a period of low and prominent left and right symmetry. And this was true for a large part of the lobule CRUS1 and CRUS2, but not for a small part of medial CRUS2, where we observed the opposite. So that we also optogenetically stimulated the cerebellar fibers coming from the inferior olive to mimic the complex spike activity. And by activating the climbing fiber, we were able to induce either symmetric or asymmetric whisker movement, depending on the area that we stimulated. So the areas in which we observed correlation between the complex spike and increased symmetric movement, they were able also to trigger symmetric movement when optogenetically stimulated. Conversely, the small area that exhibited correlation between complex spike and less symmetric movement was able to induce asymmetric movement when optogenetically stimulated. Eventually, by looking at the anatomical connection of these cerebellar areas, we found that they were differentially connected to the contralateral cerebellar hemisphere. Therefore, the main conclusion of this study is that complex spike can promote or reduce the symmetry of the left and right body movements. To have more insights into how complex spike can affect movement, I recommend reading our paper because there we also find that complex spikes are not all the same because some complex spikes can be more effective on than others depending on the level of symbol spike activity before each complex spike.